difficulty, but it seems it's resolved now. I am pleased to share research results from our group, which is about understanding the physiology, identifying biomarkers, and predicting residual feed intake in dairy cows using milk and serum metabolome data. To start with, improving competitiveness and sustainability in dairy cattle is something important. Feed cost is rising. Feed cost contributes up to 60%, and dairy cattle are implicated in environmental impact. However, the good thing is, it's possible to improve or address both issues, high feed cost and environmental impact, through selection for feed efficiency. This is one of our work. It's a table from our research. And if you can see, we compared RFI groups, that's low RFI and high RFI, for different parameters. If you focus into the dry matter intake, the low RFI cows consume an average of 21 kg, and however, the high RFI 24, and this difference is significant. And if you look at the methane production, the low RFI cows, they do emit 325, it's about 325. However, the high RFI, 393. This one, with regard to methane, the low RFI produce less methane, about 16% less. And with dry matter intake, it's about 13% less. Now we asked two questions. What metabolic adaptation of the low RFI cows enabled them to produce, to consume less methane, to consume less dry matter and emit less methane while producing the same milk production? You can see the milk production is the same and it's not significant. The second question was, okay, if there are some adjustments in metabolism or in the physiology, can you use those metabolic drivers, those physiological components, as biomarkers? Or can you use these biomarkers to predict residual feed intake? Because residual feed intake is a hard to measure trait. And with, as I said, residual feed intake is a little bit complex trait, and it requires the correct appraisal of the requirement and the intake. That means the dry matter intake, maintenance energy requirement, milk production energy requirement, and body reserve mobilization. Therefore, trying to identify biomarkers and predicting individual RFI phenotypes would be a desired alternative. For this approach, we used a systems biology approach, which includes multi-omics as a component. If you look at the first illustration, you have the genome and the phenotype. The information transfer from genome to phenotype includes, excuse me? Can you hear me?
Yeah, it's a little echoey. Maybe try again.
Okay.
Yeah, sounds good.
How about now?
Yes, perfect.
Okay. Maybe. Is it okay, Kristen, now?
Yes, looks good.
Okay, so if you see the first illustration, you have the genotype and phenotype. The information transfer from genotype to phenotype has multi-components, which we can enumerate as the epigenome part, transcriptomics, proteomics, metabolomics, and the microbiome. And these components do interact for to create or implicated in the variation of the phenotype. In our specific situation, we have residual feed intake, and we have genomics work, proteomics work, and metabolomics. What we want to do is we want to understand, we want to have a better insight about the physiology, and we want to identify good markers of metabolomics, proteomics, which can distinguish RFI groups or which can be useful in prediction of RFI phenotypes. So we laid out our experiment as during the lactation period. As you can see it, you have from calving to 240 days. We have 3 time points, and we recorded all the data that's required for feed efficiency calculation on 72 cows. And we collected milk and serum samples at 3 time points. That's 50 days in milk, 150 days in milk, and 240 days in milk. And after that, we identified the extreme groups, the high RFI, the low RFI, at all the 3 time points. And then we compared the metabolome and as well as the proteome, but the proteome is not in this presentation, between the high and the low RFI groups at all the 3 time points. Now, when we consider the lactation period in dairy cows, it's more dynamic, diverse, and it includes the interaction of different components. This includes, if you see the first graph, the first line, the top line, the milk production energy requirement, and this one is the feed energy, which is the actual energy intake. And as you can see it, the dry matter intake peaks later than the milk production. And then we of the cow always, not always, but high milk producing cows, they have deficit. And this deficit could be compensated by the body reserve mobilization, which is the body reserve mobilization decreases during the early lactation and then starts picking up. And if you consider the late lactation, we have all the milk production declining and the body reserve store increasing, and which could be a surplus. So when we set our experiment, we try to consider this dynamics, this diversity during lactation, and we set, like as I said, at 50, 150, and 240 days. And we calculated RFI, identified the extreme groups, the negative RFI, I mean the low RFI and the high RFI, and we tested at each time points. Are they different? And what we observed is that both groups, the low RFI, the one with the red line is the high RFI, the blue line is the low RFI, and they were significantly different at all the 3 time points. Now, we calculated RFI. To calculate an RFI, it is, as the definition, it's a difference between the actual and the expected. The actual, we calculated actual energy intake, as you can see it here in the equation, and we calculated also the metabolic body weight, milk production energy requirement, and MPT body weight change. The MPT body weight change, we included it to account for body reserve mobilization, and the error is the residual feed intake. And as the comparison is at the 3 time points, and we included fixed effects, parity, month year, age at first calving, and health status. So part of the milk metabolome data has been published, and for the details you can refer to this paper. So coming to the results for the milk metabolome, we did first the principal component analysis to see, like, we use targeted metabolomics that has about 118 metabolomes, 118 metabolites quantified at all the 3 time points. And we did principal component analysis, as you can see, all the 3 time points, the 150, this one is 150, and this one is the I mean, this is 50, 150, and 240. So they are all separated. It's not surprising given the lactation period is quite diverse or quite dynamic. Now, as I said, we compared we compared the RFI groups for all 118 metabolites quantified at all the 3 time points. If you look at the early lactation, we, out of 118, 16 were significantly different among the groups. And out of the 16, the major ones here are the acyl carnitines. The acyl carnitines at early lactation that increased in high RFI groups. And the increase in different research has shown that whenever acyl carnitines are, especially the medium and long chain acyl carnitines, are markers of incomplete fat oxidation, incomplete beta oxidation, which is an indicator of the mitochondrial dysfunction. That means it's possible to suggest that the low RFI cows, they do have the metabolic flexibility or the metabolic capacity to mobilize, to oxidize the fat that's mobilized during the early lactation as compared to the high RFI cows. And if you see the mid-lactation, we have also other 19 metabolites which are significantly different. And the late lactation, which is at 240 days, we have different metabolites. The major ones are amino acids. Here, the amino acids are increased in high RFI compared to the low RFI, which indicates that the physiological adjustment during early lactation that makes the difference between the high and the low RFI is about fat oxidation, mainly from this study. However, it's not the same during late lactation. During late lactation, it seems that the physiological difference or the physiological adjustment between the high and the low RFI groups is based on the amino acid synthesis or protein synthesis as it's indicated by the high RFI, as it is indicated by the higher concentration of the amino acids in high RFI cows. It's also possible here to speculate. The low RFI cows may dilute their products to compensate the energy efficiency, to have the energy efficiency they require. So we also used the metabolite lists that are significantly different between the groups at early lactation, mid lactation, and late lactation, and try to see the pathways that are enriched. And at early lactation, we have 2 significant pathways. That's the TCA cycle pathway and the glycerophospholipid pathway. However, in mid and late lactation, we can see there is a shift. So the shift is that the pathways affected by the RFI grouping are more of the amino acid synthesis. And again, we did the PLSD analysis at all the 3 time points separately to see if these 2 groups, the high and the low RFI groups, these are the high RFI groups and these are the low RFI groups, if they are different on the basis of on the metabolome, the milk metabolome profile. And as you can see it at all the 3 time points, the 2 groups, the high and the low RFI groups, are separated. They show clear separation. And the next point is that if these 2 groups are separated, which metabolites are responsible for separating these 2 groups? this one shows for early lactation, these are the top 15 metabolites, which are responsible for the separation for the two groups. And the same for mid and late lactation. So the next is that, can we identify biomarker? Biomarker with, when you say biomarker, biomarkers that we can tell with a specific area under the curve or the specific specificity and sensitivity of the classifier. So at early lactation, we have three metabolites. The first one is carnitine, second one acetyl-ornithine, third one is sphingomyelin. So the first metabolite distinguished with area under the curve 0.81. To explain it a little more here, area under curve ranges or varies between 0 and 1. So 0 means that classifier or that biomarker doesn't work at all. It just mismatches. In this case, it classifies the lower FI as higher FI, the higher FI as lower FI. However, if the area under the curve is 1, that biomarker clearly separates the lower FI and the higher FI 100%. So we can tell these ones are pretty decent. And the same at mid-lactation. We have carnitine comes first, and then we have histidine, we have creatinine. More or less, it's the same utility. And at late lactation, we have more amino acids. Phenylalanine with 0.85 area under the curve. So for the utility of the biomarker, depending on the AUC, if a biomarker is between 0.9 and 1, it's a good one. It's an excellent one. It's the best one to have. If it is between 0.8 and 0.9, it's good. 0.7, 0.8, it's fair, but less than that, it's just not of practical utility, because area under the curve 0.5 means it's just random. So now these are the biomarkers that can tell us, okay, I have this metabolite or this biomarker, and it's possible to tell if a cow is higher FI or lower FI. It literally tells the group. It doesn't tell the RFI value, what RFI value that cow has. Therefore, in this one, what we try to do here is we try to predict RFI from these markers. So we used a multiple linear regression model, and at early lactation, from 6 metabolites, we can predict RFI with a 0.76 accuracy. And we did also the leave one out cross validation, and you can see it is like 0.65. At mid lactation, 5 metabolites, with 5 metabolites, it's possible to predict RFI with 0.53 accuracy, and it's a little bit lower than the early and late. The late lactation seems it's stronger as comparable at the early lactation. So this is a very good result given prediction of RFI from other, like from milk spectra, has very low accuracy. In our group, we tried to predict RFI using milk spectra. What we got was really very low accuracy. And also some literature review, like the one from Shetty et al. 2017, they reported accuracy of 0.9. I mean 0.09. And A recent study also attempted to predict RFI from sensor data and a couple of, I think, 4 metabolites, and the result wasn't good. It was less than 0.01. It was very poor. So these ones, I can say they do work really well because the predictors are the metabolites, and these metabolites are part of the physiology or byproduct in the physiological system, and they could reflect the RFI as compared to the milk spectra or the other attempts. So coming to the second part, we did the same for serum. We compared this. We compared low and high RFI groups at mid. That's at early. That's 50, mid and late lactation stage, and at early lactation stage, these metabolites were significantly different between groups, more or less similar to the milk metabolome. The carnitine comes first, which tells still it is about the mitochondrial function, and it's about its fat oxidation during early lactation. And the second, this is the list for the list for metabolites that are significantly different between the high and low RFI groups at mid-lactation stage. And third, this one. One important thing that we observed, we try to work out like a biomarker that works across the board, a biomarker or a metabolite that could distinguish the high and the low RFI groups at 50, 150, and 240. We couldn't see that, but when you compare it with at each time points with the univariate analysis, we observed it's only betaine which is different, consistently different across the 3 time points, and it is higher in high RFI cows compared to the low RFI cows. This is also one of the metabolites. It's not significantly different at early lactation, but it's different at 150 and 240. And it's one of the biomarkers. So the plan for this one was, again, we wanted to see if we can find better metabolites from the serum. And we did the PLSD analysis. As you can see it at day 50, yes, the high and low RFI cows are significantly separated, and at mid-lactation, at 150 days in milk, they are forming 2 different groups, but still not the same as 150 and 241. And these are the list of metabolites which are responsible for the separation. And you can see it here are the carnitines and hydroxyhippuric acid, and most of them at the early lactation are carnitines, and it comes with here amino acids, and again here are short chain carnitine and the amino acids. And similar to the milk metabolome data, we wanted to see if we can find a different result. And with the serum one, at the early lactation, these are the top 3 biomarkers or metabolites. So the first one is the p-hydroxyhippuric acid, with the area under the curve 0.82. And the others are carnitins, which are very close to each other in their utility as a biomarker. At the mid-lactation, we have the acetyl-ornithine comes first, and which is an excellent marker here. It's just 0.93. And the second one is sphingomyelin and lysophosphatid choline. They are more or less comparable. At late lactation, again, acetyl-ornithine outperformed the rest, and it's commonly at the mid and the late lactation. So if we compare the serum and the milk biomarkers, in the serum biomarkers, we have a little bit more stronger, but the result seems more or less comparable. So similarly, we did multiple linear regression analysis, and at the early lactation, we have found we have like 9 metabolites could predict RFI with this much accuracy, 0.78, and mid is about 10 metabolites with accuracy of 0.76, and late lactation, we have a little fewer, but the accuracy is more or less comparable at each of the time points. And we also did the leave one out cross-validation, and the accuracy is still pretty decent. So here in this analysis, we have 9 metabolites which predict RFI with accuracy 7.8. And we wanted to know the relative contribution of each 9 of them, and the same for the mid and late, to harness this variation. So the first one, betaine contributes about 39% of the variation in RFI. And however, the contribution of betaine a little bit diminished, not a little bit, much more diminished at the mid-lactation, and it was 7%, but acetyl-ornithine has much, and also methylhistidine is also very significant in the second one. And interestingly, in the third, in the late lactation, again, we can see betaine as the significant contributor to the variation. So in all the 3 cases, I would say one metabolite stands out, and that could be with persistency across lactation, and that could be betaine. So that was the, let me summarize whatever I said so far. From this analysis, from this study, we are able to identify biomarkers that can distinguish feed-efficient and non-efficient lactating cows. And also, we are able to predict, we are able to identify a group of metabolites or set of metabolites that can predict RFI, the phenotypes. And also, we explored the physiology of the residual feed intake. And this project has been funded by Alberta Agriculture and Forestry. And also, research is always a teamwork. I would like to acknowledge Livestock Gentic and the leader. And the analysis was done at the Metabolomics Innovation Center, University of Alberta. Finally, thank you. Hello?
Great, thank you, Dr. Hailemariam. So now we have some time for questions. So if anyone has any questions or comments, you can raise your hands and turn on your microphones. or if you prefer, you can put your question into the chat and I can read it out. I did notice there are some questions in the chat already from John. He was wondering, how was the cross-validation conducted?
Yeah, the cross-validation was conducted, as I said, it was leave one out cross-validation. So we have fewer animals. What we can do is only to go for the leave one out cross-validation, and we didn't do like we couldn't have a training set and the validation set.
Okay, and can you comment on the basic biochemical mechanisms that explain differences in metabolism or metabolites between high and low RFI animals? For example, less efficient biogenesis of energy in the mitochondria due to leaky cell membranes.
Yes, as I said, especially in the early lactation, the carnitines, the medium and long chain carnitines had been significantly different between the two groups. And the concentration of these carnitines was elevated in high RFI cows, which means carnitines are these metabolites which shuttle the long and medium chain fats towards the mitochondria. And it's established that in other models, like in mouse model or in human, it's confirmed that carnitines, elevated concentration of carnitines in serum is a marker of compromised fat oxidation. That means because low RFI cows, they do have a lower concentration of those carnitines, means they do have a better mitochondrial efficiency. And not necessarily only the efficiency, the adaptability could be like the low RFI cows could possibly have more higher number of mitochondria per cell. I think this has been studied. Yes, there is one study from Germany, from the Dummelsdorf group, who they analyzed the number of mitochondria per cell in the liver cells between high and low RFI cows, and they reported low RFI cows, they have higher number as compared to the low RFI cows. Therefore, the interaction with the physiology, the role with the physiology depends on the stage of lactation. This is early lactation. During early lactation, most cows, they do mobilize fats, body fats, because they are in the negative energy balance. Therefore, the amount of fat mobilized during this time should be efficiently oxidized. It is more efficiently oxidized in low RFI as compared to the high RFI. However, the story different during late lactation, where we see its amino acids are elevated in high RFI cows and they are lower in low RFI cows. That means the low RFI cows might use the amino acids as the source of energy. Most of the amino acids in serum that we don't detect them in the serum because they might be oxidized to produce energy.
Okay, that's interesting. Do we have any audience questions? Flavia? No, I can't hear you, Flavia.
Can you hear now?
There we go. Yep.
Okay, I changed the microphone. Thanks, Dan, for the presentation. I have a question about the validation. So you started with more or less 100 milk metabolites, right?
Yes.
That you identified in 40 cows? So a group of cows, right? And
yes.
then based on those metabolites, you came up with a shorter list of metabolites that discriminated the two groups.
Yes.
And then in the second stage of research, you try to predict use the metabolites to predict the feed efficiency.
Yeah.
So, but you used exactly the same animals, right?
Yeah, that's one point. Yes.
So let me just finish
good.
my reasoning. So then you use exactly the same animals. So when you did the cross validation, you are trying to predict an unseen animal that was the one left out, but that unseen animal was used to select the group of metabolites before. So because of this, you increase the ability of prediction, right? So your cross-validation you expect to be higher because you are using the same animal that is out that was used to pre-select the group of metabolites. Right?
Yeah. I understand. I agree what you said, and yes, we wish we have external validation.
Yeah, but even with your data, you could do when you pre-select the metabolites, let's say, based on your PCA, you could out of the 20 animals leave like 5 in each group out. Completely out. Then you do your PCA, get the group of 8 metabolites, and then you do the validation, right? Using those 5 that were never used in any phase of the selection. Then you would have an independent validation. Right? So that would give a better idea of the ability to predict a really unseen, unused animal.
Yes, that's very true. That's I agree what you mentioned, and as I said, for this purpose, what we did with the validation is from same animals as you mentioned it. The reason that we didn't go to have independent animals is we didn't want to decrease the number. But that aspect is not included in the analysis.
Okay, thanks, Dan.
You're welcome. Yeah.
Thanks, Flavio. Any other audience questions? I was wondering, for your RFI prediction, I was wondering, did you try any other types of models? Like I know machine learning is popular now for trait prediction. I was wondering if you tried any other models to maybe increase the accuracy of prediction, maybe?
Yeah, we didn't try other algorithms, like whatever we use is the multiple linear regression.
Yeah.
But that's a possibility, yes.
Yeah.
And maybe the reason that we didn't, compared to the whatever so far reported, the prediction we saw is relatively good, and but still there is a room to improve it. Yeah. And that's one aspect to try.
Okay, interesting. And then I know you looked at milk and serum. Would there be a benefit of one or the other in terms of implementation, if you want to?
Yes. I prefer milk because milk is non-invasively obtained. you can easily collect milk samples. But with serum, it's less invasive, but it's less, I would say, less convenient. So if it works, it's just refining more the metabolites or the prediction, whatever we have in milk will be better than in serum with regard to the utility.
Yeah, okay, that makes sense. And any other questions? I think we have time for another question if anyone has anything. No? All right, well, thank you, Dr. Hailemariam, for the very great presentation, very nice results. And thank you for everyone that joined us today. We'll see everybody next week.
Thank you for having me.
Thank you.
Yeah.